Welcome to another edition of Research Connection, featuring today Dr. Chris Dekaney. Thanks for joining us, Dr. Dekaney.
My pleasure.
So tell us a little bit about yourself.
I went to Texas A&M University where I received my bachelor's, master's, and PhD. And during that time I studied intestinal development, the ability of fetal intestine to absorb amino acids, and so that's where I got my start in the GI tract. From there I went to Baylor College of Medicine, did a postdoc, where we focused on isolating intestinal stem cells, which is kind of where my current research focus got started. And then I moved from there, from Baylor to University of North Carolina, Chapel Hill as an assistant professor, and was there for about 8 years. And then I got here in 2015.
So microbiome.
A little bit of microbiome, mostly intestinal epithelial cell biology.
Okay. And do you have any grants related to that?
Yeah, so I have an R1 right now that's focused on the role of cells. So cells are epithelial cells that live at the base of intestinal crypts, so they're associated with intestinal stem cells, which is why we really got interested in them. Classically, cells are thought to play a role in manipulating the intestinal microbiome, but they more recently have been shown to play a role in defining the intestinal stem cell niche. So that's really where we have gotten interested in cells and what role are they playing in the intestinal stem cell niche, more specifically associated with damage and repair. So that's really what we study, is how the intestine is able to repair itself, and then how do cells, what role do they play in that repair?
Interesting. And do you have any collaborations?
Yeah, so one collaboration I have here is with Dr. Liara Gonzalez. So we're both interested in intestinal stem cells. She has a slightly different injury model with the ischemia reperfusion, and just the way that the injury comes about in the intestinal mucosa is very different than what we see. So we're interested in kind of comparing their mode of injury versus ours, and also trying to kind of cross over. We do mostly rodent mouse work, trying to cross over into the porcine world and expand our model systems a little bit there. And then I also have a collaboration with Ian Carroll at UNC, looking at the microbiome, which you had brought up a little before, and then Dr. Praveen Sethupathi, who's at Cornell, looking at microRNAs and their role in cell allocation. So.
And what are you hoping? What are some ultimate goals you're hoping for?
Yeah, well, we'd really like to understand the cells within the context of the stem cell niche. And kind of going in two different ways, well, maybe three different ways. One is, what is it that they're doing to the microbiome, particularly during damage and repair? We think there are some changes that are occurring in the microbiome and in the cells during that time. We also want to know what makes a cell. So typically when we say cells, we're thinking histologically they look a certain way, but what we're starting to find is, even though certain cells look like cells, they don't always all express the same genes, and so we're trying to figure out how does a cell become a cell. And then finally, and perhaps probably the priority, is understanding that relationship between cells and the intestinal stem cell. Is there a physical interaction, so we know that these cells actually can physically touch one another, but then also is there a paracrine interaction where the cells, who are professional secretory cells, are then secreting factors that may impact the epithelium, but more specifically the intestinal stem cells.
Neat. Sounds like interesting work.
I hope so.
Yeah. Well, thanks for sharing.
My pleasure.
And best of luck.
Thank you very much.